Okay, let's get this show on the road. All right. Have a seat, please, please, please. All right. So just before I hand this over to our esteemed guests, this is a really special event where we share the CBR and biochemistry shares the honors of this symposium. Before I start, though, I just want to remind everybody that the Earl Davies Symposium, November the 16th, all are invited. It's a great day, free. Please register. There's opportunities for students, trainees, faculty, staff to present in abstracts or oral presentations. So please register at the CBR website. And I'm going to now hand this over to a face we don't see too often. So we have to sort of reel him in from his sabbatical position not far away. It's Ross McGillivray. For those who don't know him, he was the founding director of the Center for Blood Research and an esteemed biochemist in the field of all kinds of stuff, but great stuff. Anyway, here he is to take the show on the road. Ross.
Thank you. Okay, thank you, Ed. First of all, thank you all for coming to this award presentation, which is actually the 11th time that we've done this now. And we've been very fortunate this year, as well as previous years, to host Roger Page, who was Mike's father, who comes all the way from Thunder Bay and spends a few days in Vancouver in remembrance of his son, Mike. So welcome again, Roger. This event has been sponsored by the Department of Biochemistry, CBR, and Roger's family, he and his wife, Sonia, have now endowed this award so that it will stay at UBC in perpetuity. So let me just say a few words about Michael. So Mike was a graduate student in my lab. He was actually brought up in Thunder Bay, which those of you that don't know is a very cold place just north of Duluth, Minnesota, northeast, on Lake Superior. So he attended high school there, where his father was the grade 11 and grade 12 physics and chemistry teacher. Mike then went to Carleton. Oops. Oh. Carleton University, where he did a bachelor's in biochemistry, and then he came to my lab at UBC. This was when we were in the pharmacology department, and he spent 4 and a bit years doing his PhD, and here he is proudly graduating. One of the times I met Roger. He was very successful here at the time. He worked on in vitro mutagenesis of a protease, trying to convert a bacterial protease to a blood clotting factor without worrying about the immunological side of things. So he actually learned how to do X-ray crystallography and worked with Natalie to find the structure of this protein that he'd expressed. The thing about Mike, he was always very, very social. So he really enjoyed life. He was supported by Canadian Blood Services, graduate fellowships. He also enjoyed training undergraduate students. And this is Mark Bleakley. So if anybody knows Chris Bleakley in Edmonton, this is his son, who was a lost soul until he worked with Mike. And he was literally a grad student by day, and then he was a bouncer at Derby's at night to supplement his salary and his beer gut. So Mark stayed on after working with Mike and got his PhD. He went to Melbourne to do a postdoc with a plant biochemist there, who turned out to be a postdoc in Miami when I was a graduate student. So it was like a circle had been completed. And he's now working in a the biotech company in Melbourne as a research scientist. And all because of the influence Mike had on mentoring him. The thing about Mike was he loved a work-life balance, so he worked very, very hard in the lab, but we used to have Friday beer sessions, and here we are, there's some of the empties from those. He also really enjoyed watching his beloved Vancouver Canucks lose with his dog. He then went from UBC to Washington University in St. Louis, where he worked with Enrico de Serra. And people don't know Enrico. He is an incredibly harsh man. He's Italian, brilliant guy. Nobody really understands what he does, I don't think, but except for him. So Mike joined his lab, and Enrico and Mike had an incredible relationship, and Mike was very, very successful there, both in terms of publishing really solid papers as well as getting his own Heart Association money. So after that, Mike moved to the University of California, San Francisco, where he worked with Charlie Craik. And he had sort of a junior faculty position. It was sort of a mentoring position where he was under Charlie's, this is Charlie Craik here, he was under Charlie's mentorship while he developed as an independent scientist. So when he was in Charlie's lab, Mike came up with this idea of restricted interaction peptides. So the idea was that you would take protease recognition sequence, have a membrane binding sequence, and then an imaging agent on the end. And then you could inject this into animals or people, where if the drug or the compound got into cells, particular proteases would cleave this soluble peptide away according to the specificity of the protease, and then the imaging marker and the membrane binding region would just integrate into a membrane and concentrate the membrane binding imager at the place where the protease was found. And he just started to work on this when, this is an example of using this image in I think this is an MRI. And again, a very fine detail, non-invasive imaging of where proteases were actually active inside the body. So he started doing this, sorry, that's the image there. He started doing this, eventually got a patent issued in 2020. But unfortunately, Mike, at age 36, tragically went to McDonald's in Oakland and passed away, probably of an atrial fibrillation. And it was very, very sad. To try to remember Mike, I flew to Thunder Bay and attended a celebration in Thunder Bay. And we thought it would be good to remember Mike's name here by setting up a award in Mike's name. Originally, it was supported by CBR and biochemistry. And as I said, the Page family has now endowed this award. So it is my pleasure to introduce the 2023 winner, who is Melina Messing. So Melina is originally from Germany. So she did her bachelor's degree at the University of Victoria, and then she went to the University of Lund in Sweden to do a master's before coming to UBC and working with Kelly in the Biomedical Research Center. So congratulations, Melina, and I'd like to invite her to give her talk, The Ins and Outs of a Career in Immunology, a path reaching far beyond the four corners of the bench. So.
Thank you. Hi, everyone. I would like to begin by just thanking the CBR for selecting me to be this year's Michael John Page Award recipient. It is truly an honor. And of course, I would also like to thank the Page family for making this award possible. I wasn't fortunate enough to meet Michael myself, but from what you've just heard and from what I've read about him, he truly was an outstanding member of the community. And it is such an honor to be selected to even try follow in his significant footsteps that he left here at UBC and in the scientific community as a whole. And what I think is so special about this award is that it does highlight scientific excellence, but it also highlights those outside the lab extracurricular activities that are not often thought about when one thinks of a scientific career, but many scientists partake in them. And these are the activities that really do shape us in a different way and provide skills that one might not always just gain from staying in the lab. And that's what I want to highlight here today with my talk. I'm going to give both an overview of the inside the lab portion of my career, and then also, and more importantly, maybe the outside of the lab activities that have shaped me in many different ways. And so let's begin with the inside of the lab portion. I'm a trained immunologist, and so I will give an overview of what that research path was like and kind of the twists and turns that one encounters when one does research in immunology. This was already introduced, so I'll briefly go over it. I'm originally from Germany and took already some twists and turns to get to where I am today. I went to Canada for my undergraduate degree, back to Sweden for my master's degree, and believe it or not, up until that point, I didn't study immunology at all. I knew it was a field, but I had never taken an immunology course or encountered it in any other way. But that changed when I came back to UBC for my final year of my master's, and then when I transitioned to my PhD studies. This is when I met these two brilliant scientists, Fabio Rossi, who was my mentor during my master, and then Kelly McNagny, who became my mentor during my PhD studies. And they kind of threw me off the deep end right into studying the immune system by setting me up with using this particular machine called mass cytometry, or also called CyTOF. And at the time when I first came to UBC, this was a fairly novel technology. And it, for the first time, enabled the characterization of the immune system in a high-dimensional way. And it does this by allowing for up to 40 antibodies or cell markers to be included in one panel. And then, to skip some of the technical steps, it really relies at the end of the day on conventional mass spec to produce these really distinct signals for each of these markers, and then allows you to use so many in one antibody panel. And the kind of result you get from this is shown here in this UMAP clustering plot, where using my personally designed 4D antibody panel, I explored immune population, in this case here in peripheral blood. And you can see there are the broad categories visible, but there's also so much detail contained within each of these populations. And it's a really powerful tool, particularly to perform exploratory analysis when you don't really know yet what you're specifically looking for. And so this particular tool I used for a few different projects that I would like to highlight. At least I'm going to highlight two out of them here today. So first, when I began my PhD studies in Kelly McNagny's lab, he had come up with this super interesting project that I was intrigued by immediately. But before I actually talk about it, I just want to acknowledge the members that have worked with me on this project and are still co-leading this project with me today, Kevin Leslie and Samuel Shen. And so at the time, Kelly had set up this collaboration with the Child Study. So this is a study that over 10 years ago now enrolled over 3,000 children and their parents into the study. And their overarching goal was to figure out what drives the development of allergic disease and other inflammatory childhood conditions. And so the study followed these children throughout their life or their early childhood, since before they were born all the way until now, and they're now over 10 years of age now. And throughout this time period, the child study collected, of course, a vast amount of clinical data and performed various disease diagnoses, and they also obtained a vast amount of biological samples. And one type of biological samples that they collected was actually the isolated immune cells at a time of birth, so from the umbilical cord blood. And they archived those immune cells, and that was then available to us at the time. And so now we have this exploratory tool that allows us to study the immune system in a lot of detail. And we have these archived umbilical cord blood samples from children that have been throughout their childhood either have remained healthy or have been diagnosed with various forms of allergic disease. And so Kelly came up with this really, really interesting question. Can we actually look at the immune system at time of birth in the umbilical cord blood when the child hasn't been exposed yet to any outside influence yet, but everything that has happened to it was coming solely from the mother? And can we find immunological signatures that can predict if a child will go on to either remain healthy or develop allergic disease later in life? And of course, if we were to find a signature like this, then that would mean that the child was predisposed to develop allergic disease during pregnancy and not to counter conventional belief by postnatal influences. And to address these questions, actually very simple set out of the study, we requested these archived umbilical cord blood samples from different groups, children that had either remained healthy or had been diagnosed with various forms of allergic disease. And then we designed our antibody panel to study the immune system in these samples. And now you're probably on the edge of your seat wanting to know if we did find those predictive signatures. And as fate had it, at the time when we were just ready to go undertaking this the study, we had the samples in the freezer, the tool was developed. This is when, unfortunately, we had to stop all research because of the COVID-19 pandemic in 2020. But being in Kelly's lab, that means that immediately new collaborations are formed and new ideas for projects are right around the corner. And so instead of just waiting the pandemic out, we actually decided to actively participate in it, because now we have this exploratory tool that is ready to go, and there is a virus that is also causing an immune response that deserves to be studied. And that's exactly what we did. We shifted our project not to the study of immune cells in cord blood, but instead to the immune cells in peripheral blood of COVID-19 patients. And this is just a snapshot of the kind of data that we generated using our tool, which is this really detailed heat map that you can see here, all my 40 antibodies describing the immune system in the peripheral blood samples of COVID-19 patients, and then the associated clustering plot showing all of the different immune populations that you might expect in peripheral blood. And for the interest of time today, because I do want to also talk about my extracurricular activities, I will make a long story very short and tell you what we found. So using this high-dimensional tool, together with also the analysis of excreted proteins in the peripheral blood, we found a biomarker signature. And we found this biomarker signature specifically in the peripheral blood of ICU COVID-19 patients. And the peripheral blood was given to us right when they were admitted to the hospital. And then the signature actually allowed us to separate the COVID-19 patients into two different groups. We were able, by just looking at the signature, to see which patient would go on to stay in the ICU for a short amount of time and go home and fully recover, from those patients who actually ended up staying either in the ICU for an extended period of time and had a very challenging time in the ICU and or passed away from COVID-19. And so a predictive signature like this that can tell you disease outcome right at when these patients are admitted to the ICU can of course have powerful consequences for healthcare, especially in a time when resources are short and it is a novel condition and it's uncertain what type of medications are useful and might impact a patient's life. It is very useful if a clinician has a tool to triage patients and provide more personalized, effective care. And that was, of course, a very brief summary of this quite intensive project. But if you're interested in reading more about it, it is published in Frontiers. So this is actually all I'm going to tell you about my the inside of the lab aspect of my career. But I hope I've shown you that the career of an immunologist can be very versatile and can be adjusted depending on the need. And this was actually a fantastic experience being able to participate in research for an ongoing emergency and really make a difference. So now I would like to shift my talk to the extracurricular outside of the lab portion of my career, which I think is just as important and has definitely given me skills that I treasure on a daily basis. And the three aspects I would like to highlight are EDI advocacy, science outreach, and mentorship. So I'll begin with the EDI advocacy. This is actually one of the more recent activities that I started to take part in. But of course, EDI, equity and diversity and inclusion, is a hot topic in science, and so it should be. It is definitely time that we have an equal space in science, and so it is a great opportunity to partake in creating that equality. And I did this by joining the EDI committee of the Canadian Society of Immunology. The Canadian Society of Immunology is a society that aims to bring together all Canadian immunologists and provide a platform for them to exchange research and network and in generally stay in contact in such a big country, across such a big country. And the EDI committee was formed to foster EDI within the society, and it's actually led by Janet Valdez and is, I think, now 12 members strong. And right away when I joined, I only joined last fall, but right away when I joined, I was able to participate in this really interesting research of the historical records of the Canadian Society of Immunology. We specifically aimed to understand the gender representation within those records to see where does CSI stand both historically and today with regards to EDI. And we analyzed a number of different categories, including CSI leadership and CSI award winners and annual meeting participants. But I just want to give you just a highlight of one of these categories to show you the type of trends that we found. And I think the analysis of the award categories really nicely demonstrates what we were able to uncover. So these are two awards here that are given by the Canadian Society of Immunology, the CINEDA Award, and then a more recent award that is aimed at new investigators that are early in their career. And perhaps not surprising when you look at the historically at the award recipients for the award that dates back to 1987, for many, many years, it was, of course, only men that received this award because the science community was dominated by men at the time. But you do see this shift towards equality in later years, especially since the year 2017. And that was very encouraging to see, and we're hoping that this trend continues. But then, unfortunately, looking at this more recent award, where you would kind of expect a more equal representation of both men and women recipients, we still see a bias. And there doesn't seem to be much of a trend towards equal selection, perhaps in the last few years, but we have to see if that actually keeps going. And that was surprising to us. Why would this award perhaps be still be biased towards the selection of men? And one of the many explanations that we could think of is perhaps women right at that time of their career, when they're early investigators, that's when they go also often on maternity leave. And that's when they, on paper, perhaps look that they're like they're less productive. And so this is just one example of the many things that we think the CSI can improve on when selecting these award recipients, and to address many of the other biases that we found analyzing this data set. But in a short year, I was able to really uncover some really, I think, what will be impactful findings, because it will educate the society on how to move forward in a more inclusive manner. And this is actually submitted this week, and I'm the first author on that manuscript. So now let's move to science outreach, also another very important aspect, I think, of science. And the way I participate in a science outreach is by joining training committees of scientific clusters. In this case, the immunotherapeutics research cluster that was actually founded in 2022. And that is a cluster established in British Columbia to bring together researchers from, or immunologists from all different sectors, health policy and immunologists in industry, researchers, clinicians, to foster communication, to foster this community, and to drive the translation of immunotherapeutics. And as part of the trainee committee, I'm actually the co-chair of the trainee committee together with Bernice Ramos. We are organizing all kinds of events that are shaping the immunology space here in British Columbia, that allow for trainees to share their research and for the community to come together. And just some advertisement here at this point. One of these events is this work in progress session that we are hosting on a bi-weekly basis, and where this is really directed at trainees to share research that is in progress and get valuable feedback from experts in the community. And so if you are interested in presenting your research, we still have some open slots for this fall, so feel free to contact me. Another really exciting outreach event that I recently was able to take part in as a member of the Immunity Trainee Committee was an outreach event that was actually held at Science World here in Vancouver on the Day of Immunology. And this was to celebrate immunology and to show the general public the impact that immunology can have on the community. And so we had a panel of experts that where the general public was able to ask a lot of questions. There were also many young members in the audience that had fantastic questions. And then there were a lot of immunology-themed games that really showed, especially the younger members, how what immunology is and how it impacts society. So this was just another example of this type of science outreach that I really cherish, and that I think is really important to show the public what we do on a daily basis as immunologists. So finally, I also want to touch on mentorship, which might be one of the most important extracurricular activities. Mentorship does come naturally in the lab, of course. When the more senior you get, and young students come into the lab, naturally you start mentoring them. But that's, of course, when students have already decided they're interested in research, and they're there because they're interested. So you obviously teach them techniques and you mentor them maybe through grad school, but you don't, you're not reaching the roots, if you can call it this. But there are other programs that outside of the lab we can participate in to mentor even members that haven't found a lab, that they don't even know yet if they're interested in science. And UBC offers this great program called the REX Mentorship Program, where you can sign up as a mentor and you get matched with pretty much as many mentees as you would like. And you guide them through a research project and at the end, a research presentation. And these mentees that join this program have never entered a lab, have no research experience. They just know they might be interested in science. And so as a mentor, you kind of get to inspire them, show them what research is like, and maybe out of the many mentees you get there, if you even inspire one or two just to become scientists, then I think you've done your job. And it was, this is what I participated in last year. And this is me with my 4 mentees at their final poster. It was a very proud mentor moment for me. And one of them still to this day has stayed on with me in the lab and is still my student. And so I think that shows that they had a good time doing this research project, and that at least one of them maybe will become a scientist at the end. So before I conclude my talk, I do want to touch on one extracurricular activity that is way outside of science. But since Michael also seemed to have enjoyed many of these activities, I did want to touch on it. I'm not really that much into sports, but my outside the lab activity is joining local orchestras as a cellist. This is a picture from one of the orchestras I'm in, the West Coast Symphony. We played at the Orpheum last year, and we had full choir and everything. And it was a fantastic experience, and it really gives you a nice cultural break from science. And it's also an event where I can invite people to. A lot of my colleagues come to these events, and I think it shapes the community also outside the lab. And to tag onto this, we have a concert coming up if you want to come on October 14th. We're playing a program called The Romantic Escape, and it's a fantastic program. And if you need also a break from science, a cultural break from whatever, you're more than welcome to come. And with this, I just would like to end with some acknowledgements. I acknowledge most of the guiding members throughout my career along the way, but I do want to highlight again Kelly for being an outstanding mentor. I think grad school is made or broken by having a good mentor, and I had one of the best. And then, of course, I want to highlight all the members of the McNagny Lab for being a great support system and great colleagues. And finally, again, thank you so much to the Page family and to the CBR. It is truly an honor to have received this award, and I just hope I can make it justice. Thank you.
Thank you, Melina.
Thank you.
Does anybody have any questions for Melina? I would like to point out that this award has gone to 5 ladies and 6 men. So we're almost there. So, Ed?
I was just saying that, how can you say that music isn't science? Oh my goodness. The science of music and music of science. They're so interesting.
That's a fair point. My apologies.
You have to stand outside the light.
It's great to be here.
So with your COVID-19 study, could you correlate those biomarkers that resulted in people staying in hospital longer with anything?
Mostly just with the outcome of the disease. That was really the biggest factor. We had a relatively small number of patients, so I think if we were to extend this to more, it would. I think it would show some really interesting findings.
Conversely, the people that didn't get bad disease.
That's the same answer, I would say. Yeah.
Okay. Yeah.
So, how does the information that you gained from that COVID study, is it being used in the clinic, or could it be?
I think it could be. It would need to go. So we basically did a discovery. There's many different steps towards discovering something to get it to the clinic. We did the discovery portion. So the next step would be to test it on a much larger set of patients and diverse cohorts around the world to see if it actually holds true for different populations, and then see which particular markers would sort of the best to be translated. But I think it would be very powerful. I think immune biomarkers are not used enough, and we have a lot of the discovery, and then along the way they end up not making it through the clinic for various different reasons. But I think if we learn how to translate them properly, I think they can make a huge difference. It's definitely something I'm thinking about a lot. How can we accelerate translation eventually? And joining something like the immunotherapeutics cluster is one way, right? We're bringing together people, we're getting people to talk, and how might we actually be able to translate these discoveries?
Melina, can you go back to your slide at the side top?
Further back?
From a non-science point of view, the art of science, we have a cover competition for our semi-annual magazine.
You want me to submit this? All right.
I am not a judge. Just a comment, suggestion.
Yes, that's a very good idea. Yes.
Okay, thank you. So I'd just like to ask Roger to say a few words before he presents the plaque to Melina. So
First of all, do I need this thing, or can you hear me in the back?
No, you need to use it because there's people online.
the people in Kamloops can't hear you.
Oh, we're online. How would I know that? Nothing says you're online. Okay, so I will use this, and then I'll tone it down a little bit. That's a little problem. Yes, I didn't really think I was going to come here this time because I think it's going to be my last trip here, but I wasn't going to say too much. And in fact, anything you want to really know of interest in Michael's case, it's on the internet, because the Discovery Fellows program in San Francisco, where he finished up his, sadly, the work that he had been doing, made a wonderful presentation and named a symposium after Michael. And that symposium under the Discovery Fellows program is now backed by $140 million. And that means if a student that goes to the university there and is getting now into their final 2 years, there's some special things that they have to have as credits for it, but they get full coverage for their last 2 years financially, and they have to project certain things into the community. Well, naming the symposium after Michael and having that wonderful background at a university that the pharmaceutical program there has ranked for years as first, second, or third in the States. And that is really a very impressive university. And again, working here when he got his postdoc degree with Ross, he had a good mentor, a good person to work with here. That worked out beautiful for him. And the same thing holds true when he was in California. The professor he had there that he was doing his research work with was an excellent man, and he's got great qualifications that received awards and so on. A fellow by the name of Dr. Kreck, and you could pull his name up and you'd find out that he's a very special man too. And on top of it, he did his research work in a building called the Genentech building there. And that's where the sad disease that so many people have, diabetic, he produced artificial insulin. And the money that he got from that created the Genentech building. So that's interesting too in its own right that he actually got to that university. Also, just to do a little bit of a finishing up thing, he passed away in 2013, and in 2015, the university decided, yes, it was time. He had a patent, and they decided that they would fund the patent and submit it to the American Academy of Patents, and it got approved. 2015, it was submitted. 2020, before it was published. It just showed you how long it takes for people doing research work for their achievements to come out. You can look at that Nobel Prize winners for that. In fact, there was something very interesting there about the Nobel Prize winner in physics, same university that Melinda was at in Sweden. And it was just announced 2 days ago. So I thought that was quite impressive to know she was working in the same university surroundings. Again, when you think of what to say under these circumstances, I've been very happy that we established this as an endowment forever. And knowing that he also has this wonderful structure in California, I can see the merits of it. Because Michael always said in terms of postdoctoral work, you know, postdoctors, when they start doing their postdoctoral work, they don't get tremendous money, so they need more. So he had commented that long before he ever passed away, because when he was doing his postdoctoral work, he would love to have had a little more money at the time. And then he had to do a lot of serious work writing to get awards, to get funds. And he achieved very well. 2 months before he passed away, he had received $100,000 from the university to carry on his work, and another $100,000 from the big what's it called, Ross? The National NIH. NIH. Yeah, and they gave him another 100,000. And he had that sum from money from them over the years. So again, my point is that I have enjoyed coming out here, and it's always a pleasure to see young people that are working hard and achieving at a high level. And I was a science teacher in the high school system, so I'm well aware of the backgrounds of it, but not the depth of knowledge. So that's about what I would like to say, and I will now pass this back to Ross. I'm going to add one other little comment, by the way. Michael's initial work was with blood clots. And I feel he got into doing work with blood clots because of me. I was an alpine skier up until I was 78 years old. Well, at his age, he skied too. I had a ski accident and I had a blood clot in my calf. And I had a doctor friend and I went to a party with him and he says, you'd better go down to emergency and get checked out. So sure enough, I did. And then I was in the hospital for 1 week on rat poison, a general term for a very famous chemical. And anyway, then the following year I said, you know, I've had all my family, I'm a traveler. My family had been over to Europe with me on occasions, but Michael had never come at his age. So when he was 6 years old, I decided, okay, I'm going to take him to Europe and we're going to go over for a period of time. And that's what led to, I'm sure, why he decided to do blood work, because we were so bonded to me. So thank you very much. Yes.